Well, thank you for that excellent presentation. It's unusual to see scientific research with such dramatic results. We have an open floor for questions. If you'd like to ask a question, please stick up your hand. I'm going to hand you the microphone.
Yeah.
You know how you said about the serum making out of your blood? What happens if you did have a disease which caused the blood to not be such a good choice? You know when you were making the serum, how you made it out of blood, and what happens if you were infected with something like HIV? What would you do then?
Unfortunately, if the patient is HIV positive, we are not allowed to take these cells in the GMP lab to grow, because then you contaminate the GMP lab, you see. That's why one of the criteria to allow me to do this kind of operation is that the patient is not infected with even hepatitis, right? Unfortunately, there's a GMP regulation. Like I said to you before, these GMP labs are designed for safety and quality. And then to make it standardized, this to make it safety for every single patient. You can see the volume, like the lab that we work, the main purpose of the lab was for blood, cell transfusion, you see. And then there is a huge number of patients going through these labs. And then obviously you can't afford to run the risk of transmit this kind of disease to somebody else. There is no evidence, hard evidence, that other than that, the serum can be not good for the patients. I mean, there are lots of been used autologous serum for treatment of patients, not necessarily just to help the cells to grow, for a number of years very successfully. And then, like I said, these are the best teardrops you can get for the patients, really, because it's close to normal. It's close because the tear is not, although the tear is just a little bit of a drop, but there are so many ingredients in the tear that you can't mimic at all. Even the serum is close, but it's not the same. Then it's kind of, I can see the controversy there. You have somebody who's blind, but has HIV, what can we do? Then that's a point for discussion.
Is there one more question?
You said that this helps with sight in like the medium to long term, like 6 or so years. Once sight starts to deteriorate again, would it be to the level that you have to go through this entire procedure again and do the stem cell transplant and a corneal transplant? Or is it because they've gotten these new stem cells, it's able to repair slightly more effectively and it's a less severe treatment the second time around?
Okay. I mean, these data is the data we have. Like I said before, the first patient we did was in 2006. Then now it's 8 years, more than 6. Then there's the medium kind of follow-up that I had for the patient. Then that we don't know. That's why you have to be so careful, consent patient. The patient needs to understand that this treatment is not well established yet. That's not the end of the road. And then we are, like I said, we keeping the patient like Newcastle patients. Although now I treat patients from all over the country, from Aberdeen to Bristol, Belfast, etc. And then so far we never lost any patient, because one of the things that we make sure they understand is get a motivation from the patient, because you want to see them for life. And then we don't have any evidence that they will necessarily disappear and stop functioning. The data from Italy is longer than mine, like I said before, and they have been doing this operation now for in excess of 10 years. And then their data suggests that the failure they had is early. When they get later on, like 10 years, they don't necessarily fail, right? Our data didn't demonstrate any early failure yet. All the patients we've done so far, now we have 30 patients done. Then we don't know the long-term survival of these cells at all, and we can't see the cells. Then we don't know, like for example, you transplant X amount of cells, and now do you have X minus what? We don't know that. Then unfortunately it's not quantifiable, and that's why we need to carry on seeing the patients. Obviously, we are hoping now that with the kind of data we have, and the medium term, like 6 years, 7 years, 8 years result, that we may be able to persuade the regulations to allow us to do this outside research. Because that's one of the words for, you have an orphan disease and you don't have a treatment for it yet. That's the treatment. Then we may be able to persuade the regulatory bodies saying, let us go out and do more patients. Although we still want to collect the data, but not as like a research can provide as a service, right? to give the opportunity for more people to have this kind of disease. Sorry, to have this kind of treatment. Right? But we don't know the long term, unfortunately. I wish I knew. Time will tell.